Host-microbe interactions. In this lecture, you'll learn more about normal microbiota, how normal microbiota and pathogenic organisms establish infection in the human body, the stages of infection which mirror the growth curve of microbes, the difference between signs and symptoms, a little about epidemiology, and nosocomial infections. The body is the perfect environment for growth of many different types of microorganisms, both normal microbiota and pathogenic organisms that cause disease. The environment of the human body has a wide variety of microenvironments that differ in temperature, pH, available nutrients, and availability of oxygen. Because of the variables among the microenvironments, each of them harbors a different set of microbes based on their needs. When the body undergoes changes, the microenvironments can also change, and this may cause the composition of the microbes within that microenvironment to also change. Recall that normal microbiota are those that live in us and on us. They are normal residents of our bodies. This table lists some of the sites that harbor normal microbiota. You should be familiar with these. You should also be aware that sites exposed to the outside, including the digestive tract, are non-sterile and contain normal microbiota and are sites where pathogens may try to gain entry to infect the body. All other places, as well as the fluids contained within, should remain sterile. You should also be familiar with the sites in this list. Note that the amniotic fluid surrounding the embryo and the growing fetus is also sterile, so the baby is contained within a sterile environment until birth. Babies get their first exposure to microbes and start to acquire their normal microbiota when the fetal membrane ruptures. As they travel through the birth canal, babies come in contact with additional microbes. 8 to 12 hours after delivery, other microbes from breathing air, feeding, and contact with parents and healthcare workers colonize the skin, GI tract, respiratory tract, and genital urinary tract of newborns. Note that the microbiota that colonizes the GI tract of bottle-fed babies differs greatly from that of breastfed babies. As babies start teething and shoving random items into their mouths, they're exposed to even more microbes, as well as when they are weaned off the bottle or breast milk and start eating solid foods. Exposure to microbes that eventually become part of the normal microbiota helps to stimulate the baby's immune system. These normal microbiota play a role in preventing pathogens from entering the body. Because the baby's immune system is still immature, exposure to pathogens should be avoided as much as possible. Skin is the largest and most accessible organ to microbiota. It harbors resident microbiota as well as transient microbes. Residents are normal microbiota. They are stable, they're very predictable, and they are less influenced by hygiene because they're firmly attached to the skin or other body surfaces. Bacteria, fungi, and mites are all part of the normal microbiota. Transients are microbes that are just passing by. Some may be pathogenic and some may not. We come in contact with transients all the time, and they're easily washed away, so their presence is greatly influenced by hygiene. The GI tract is a long hollow tube with several twists and turns. Even though it runs through the body, it's open to the outside at both ends and is a non-sterile environment with many microbiota throughout each section. Different microbiota that live within the gut have various pH and oxygen requirements and have their own little niche within the anatomy of the GI tract. The largest amounts of microbiota can be found in the GI tract. Within the oral cavity, you'll find the most diverse and unique microbes of the body. And within the oral cavity, there are several microenvironments that harbor the large variety of organisms. The large intestine contains the largest population of microbes, about 10 to the 8th to 10 to the 11th microbes per gram of feces. That's well over 100 trillion microbes per gram. The large intestine favors the growth of anaerobes as well as facultative anaerobes. Microbiota are essential to our health. They prevent infections, enhance our immune system that defends us against foreign invaders. But things like antibiotics and dietary changes and disease may alter the microenvironments and therefore alter the microbiota. Taking probiotics or eating and drinking foods that contain the types of microbes found in the normal microbiota of our gut can reintroduce them to our system and stabilize imbalances. The infection process begins with entry into the human body. The pathogen must find a way in to set up infection. These portals of entry are often the same as the portals of exit. Infectious agents enter through breaks in the skin. They enter the GI tract by ingesting infected food or water, or eating food with unwashed hands. They can be inhaled and enter the respiratory tract, and they can also enter the urogenital tract and travel upwards to cause bladder and urinary infections. Endogenous microbiota, or endogenous biota, is another term for normal microbiota. As long as these organisms remain in their own microenvironment, they don't cause problems, but they can cause infection when they are found in places where they do not normally reside. For example, E. coli is normally found at the end of the digestive tract, but if it's ingested and ends up in the small intestine, it can cause severe diarrhea and other stomach flu symptoms. Once organisms reach their portal of entry, they attach firmly using structures discussed earlier in the course, like fimbriae, capsules, surface proteins, viral spikes, and hooks. Once they attach, they enter the host, and then they use methods to avoid detection by our immune system. They must avoid phagocytosis and other human defenses. Once they reach the target tissue, they cause damage and disease in ways that are specific to each organism. For example, some organisms produce toxins and enzymes that damage host cells, causing them to lyse. Other pathogens cause the host immune response to overreact, resulting in the body damaging itself. The portals of exit are similar to the portals of entry, and pathogens generally exit in the same manner in which they entered the body. Two terms to know are true pathogens versus opportunistic pathogens. True pathogens are those that cause disease in healthy people. For example, the flu. Opportunistic pathogens cause disease in people with weakened immune system, like people with AIDS. Opportunistic pathogens usually don't cause disease in healthy people. Pathogens can originate from outside the body. These are exogenous microbes. Other pathogens already exist on or in the body. These may be normal microbiota that have invaded an area of the body where it is not normally found. This slide shows the different methods pathogens use to attach and adhere to the host. The microbial structures listed here are used to attach to specific structures on the host. For example, the fimbriae of E. coli cells attach specifically to the cells of the small intestine, as shown in this figure here, marked A. And other structures like the flagella are used to help the bacteria swim to its target tissue within the host. Some structures like capsules, pictured here in figure B, protect the microbes from destruction by our immune system. And spikes on viruses, like the coronavirus, allow attachment by binding to specific structures on the surface of host cells, as seen in figure C. This table lists a number of human pathogens, the diseases they cause, and the method in which they attach to their target cells. You should pay special attention to the pathogens outlined in red: E. coli, Streptococcus mutans, and the HIV virus. Once the pathogen has entered the body, it uses virulence factors to establish themselves and cause damage to the host. Virulence factors can be structures on the organisms, or they can be toxins produced by the organism. Some virulence factors are more damaging than others, so a pathogen's virulence factors determines the degree of damage that it will cause, as well as the severity of the disease. Virulence factors can have anti-phagocytic effects that prevent our own white blood cells from destroying them. The toxins produced can be secreted to the outside of the microbial cell into the infected tissue. These are considered exotoxins. The toxin may also be an endotoxin. These endotoxins are released after the microbe has been damaged by our immune system. An example of an endotoxin is the lipopolysaccharide of gram-negative pathogens. The 4 stages of infection and disease are the incubation period, the prodromal stage, the period of invasion, and the convalescent period. You should know each of these stages, the impact the pathogen is having on the body at each stage, and what is happening to the pathogen at each stage. The incubation period is the time of initial contact to the first signs of symptoms. It is during this time the pathogen is adapting to the host environment and traveling to the target tissue. The pathogen is just beginning to multiply. This incubation period can last several hours to several years, depending on the pathogen. The second stage is the prodromal stage. At this time, the pathogen is still multiplying and starting to cause more damage, so that the host is just starting to feel general discomfort. This stage can last 1 to 2 days. The period of invasion is the height of the infection. The pathogen has reached its target tissue and is causing the most damage to the host. The host now has signs and symptoms that are specifically associated with the pathogen, which is multiplying at very high levels within the host tissues. The convalescent period is also known as the recovery period. The host is starting to feel better, and there is a decrease in the number of pathogen cells due to the immune system. Pay attention to the pattern of the stages that highly resembles the growth curve of microbes in one of our previous lectures, where there is a lag phase, an exponential growth phase, a stationary phase, and a death phase. This table lists common signs and symptoms of infectious diseases. You should be able to distinguish between signs that are objective, measurable, and observable versus symptoms that are subjective and can only be described by the patient. Even though symptoms may seem to be measurable or observable, you have to ask yourself if you can measure or truly know for sure what the patient is experiencing without them telling you on their own. Epidemiologists are concerned with determining the how, what, where, why, and when of the acquisition and transmission of infectious agents to prevent further spread of disease and prevent future disease from recurring. Epidemiologists are trained to determine the organism causing the problem by collecting as much information as possible. Is it gram-negative? Is it gram-positive? Is it susceptible or resistant to antibiotics? And what are the virulence factors included in its pathogenicity profile? Finding out the timeline of when the pathogen was transmitted will help determine how fast it's spreading and how fast people need to act to prevent further spread. It's also important to know where the pathogen originated so others won't come in contact with the pathogen. Knowing how it's transmitted is also important to slow the spread. Is it a communicable infection, spread from person to person? Is it transmitted directly, meaning do you have to have some direct contact with the infected person to contract the infection? Or is it transmitted through air? All of these things must be taken into account. We can encounter pathogens in their natural habitat or reservoir, for example, by drinking contaminated water from a lake or swimming in that contaminated lake. Pathogens can also be acquired through contact with infected individuals or contaminated objects. This often happens in hospital settings. Carriers exist in and out of hospital settings. These people inconspicuously shelter a pathogen and spread it to others. They may or may not have symptoms. Asymptomatic passive carriers are people with no noticeable disease signs or symptoms who harbor infectious agents. These people can spread the infections to others unknowingly. Convalescent carriers are those who have recovered from their illness but remain capable of transmitting to others. And chronic carriers are those who continue to harbor a pathogen for months or even years after the initial infection. Nosocomial infections are acquired during a hospital stay, usually during surgical procedures from equipment used or from medical personnel. Nosocomial infections can be life-threatening if the pathogenic strains are resistant to antibiotics or if the pathogens are highly virulent. But these infections can be prevented. Medical aseptic practices like proper hand washing, using disinfectants to clean areas that the patient may come in contact with, and isolating highly contagious people from the general hospital population can lower the microbial load on patients, caregivers, and the hospital environment. Surgical aseptic practices like proper sterilization of surgical equipment, using disposable dressings and clothing, filtering the air, and proper disinfection of surfaces provides a sterile surgical environment to keep the patient safe and free of infection. Use the last two summary slides to guide you as you study this material. This is the end of this lecture, and thank you for listening.